Okay, so for this topic, I want to cover just really briefly some of the basics of molecular and cellular biology. I think this is a really important topic. However, I'm not going to go into a ton of detail, but this course, when I took this at the University of Arizona, literally changed my life. It blew my mind. Um, we went into a lot of detail, but I mean, this basically explained why everything works the way it does. And so I'm going to try to hit on some of the more poignant basics, and in another video, really dive into transcription and translation, which is, I think, probably the most important part of this to understanding any of this biology. And so, just to start off at the very basics here, um, so what you're seeing here is from Darwin. Everything that I present is in the context of evolution. And so irrespective of any religious beliefs, evolution is a biological fact at this point. We have so much evidence to support that it has occurred and that it continues to occur. And so I don't think that means you can't have religious beliefs, but this process which we observe we could do it in the lab. I'll even post a link to a video which demonstrates antibiotic resistance and the evolution of this within a 5-minute video. And so everything is in light of evolution, and it makes sense in light of evolution. And so what you're seeing here on the left is just sort of Darwin's early scribblings and descriptions of how he believed organisms were connected through this descent with modification. I'm not off the top of my head familiar with the whole history. I know that he did not come up with this idea on his own. I think his uncle or somebody else close to him sort of had this idea of rolling, but he was the one who really laid a clear-cut foundation for the rules that it should apply if it were true. And then he went out and tested those in the field, and he saw that they were true, and we continue to see that today. And so we are able to, through the advent of molecular technology, we can sequence DNA and trace this lineage to every organism on the planet. And we know from this that they all arise from this common ancestor that's depicted here at this very root of the tree. And so every living thing on this planet can be traced back to every other living thing. And there was this common ancestor that links them all. Now, broadly, we break these categories into what we call prokaryotes, and these are a type of single-cell organism which lacks a nucleus, and we have eukaryotes, which can be single-celled, but we usually think of them more commonly as the multicellular life, but they can be single-celled, and these are defined by having a nucleus, which I'll cover on the next slide. But so we see that there's these 3 domains, what we call domains of life. Eukarya, which houses fungi, plants, animals. We fall into here. We have the archaeans, so archaea is here, and then bacteria is separate over here. And so where did viruses sort of fit into this? It's kind of interesting. When we look at their genomic material and we trace back their lineage, they don't fall into this category where other life does, and we can't trace them back to one single common ancestor. And so I've just provided 2 examples of the different types, the broad classes of viruses, so single-stranded RNA, and these are DNA viruses. And we can see here by doing, these are called phylogenetic studies, or you're looking at a phylogeny, which is basically a tree of life. When you're looking at just the DNA viruses, we have maybe this tree here, which we can root and sort of find this commonality between these. But when we look at the papillomaviruses, the viruses in Adenoviridae, they don't have a relationship to these other viruses here. And so that's kind of interesting. We can't trace back their lineage. And even further, we have this other class of viruses, one of like 7 or 8 different classes of viruses, and these group separately from the DNA viruses. So it's a really interesting question about where did viruses come from, and are they part of what we consider life? And I think those can be debated. They are debated. Viruses, the current thinking in the field is that viruses probably were a cell that lost through evolution some of its components that it required to exist on its own. And so now that it's lost those, it's sort of taken on this parasitic nature where it has to depend on another cell. Um, Parasitism is like the most abundant form of life on this planet, and so I don't think that that's that surprising that if you can get somebody else to do what you need done, or something else, then that's a nice way to go about living. Um, and so, just to that point on life, we don't necessarily consider viruses alive. They exist in sort of two phases. They exist outside of a host cell, in which they carry out no metabolic activity. So they are not doing any of the life processes that we associate with life on their own until they get into a host cell. And from that point, whether it's a eukaryotic host or a prokaryotic host, they are stealing, robbing, subverting, doing everything they can to take what they need from that host to carry out their functions. And from there, they can replicate and they produce proteins and they evolve. And so they do exhibit sort of properties and characteristics of life. But rather than debating whether or not they're alive or not alive, we just refer to them as acellular. So they're, we talk about cellular life, and that is everything but viruses. Um, yeah, so that's where they fit in the big scheme of things. So getting more in depth with the cell here, this is a cartoon of the eukaryotic cell, and this is a really densely packed structure, and it houses a lot of things. And overall, the main point of the cell is to basically create a barrier between the outside of its environment and what it's doing on the inside. And so it does this through the use of a plasma membrane, which is this thing that surrounds the outside here. So I just want to cover some of the major components of a cell that you would maybe hear about when somebody's talking about immunology or virology. And so when we're talking about eukaryotic cells, the defining feature of a eukaryote is that it has a nucleus, and that is this large structure here. The nucleus houses the DNA of the or of that cell. The endoplasmic reticulum is this large membranous lipid membrane structure. It's contiguous with that nucleus, and it's the site of lipid synthesis. You have protein synthesis for destined proteins that are destined for a secretory pathway are synthesized in here. And so it serves those functions, among others. We have the Golgi complex, which is this green structure here, and this can act as sort of a sorting station. You have vesicles that bud off from this ER, it moves to the Golgi, and that moves out of the cell, and that system works in reverse, and it also has a few points where it branches off to send things to other places in the cell. Let's see, what do I have here? I can't really read that. Oh, mitochondria. Sorry, it's kind of light out here. So mitochondria, these are these orange-looking structures here. These themselves are believed to at once have been their own prokaryotic bacteriums, and they've evolved to supply the cell with energy. So your ATP synthesis, your ATP is the really the energy sustaining molecule of life, and so that's produced here in this membrane of the mitochondrion, and so these are really important, obviously, for that reason. Um, let's see here. Sorry, I'm having a difficult time reading my screen. Okay, and so we have the other important things here. Then would be the cytoplasm, which is all of the space that is within the cell but not in the nucleus. So the space between the nucleus and the plasma membrane, which I've mentioned, is this surrounding lipid envelope or lipid layer, bilayer. And then the cytosol is is another name, I guess you would hear the cytosol be called. Sorry, you would hear the cytoplasm be called the cytosol. One is really the space, and the other is more of like the jelly-like fluid which fills that space. But they're sort of used synonymously. Okay, so the last thing I want to cover for this little video is sort of what the nature of DNA is, and this was work that was done by many scientists, but most notably back in the 50s here, 1953, Watson and Crick and Rosalind Franklin were all instrumental in sort of explaining the chemical nature and how DNA works at a molecular level. And so we know that it is a right-handed, double-handed helix, and it consists of these four bases, which are adenine, thymine, cytosine, and guanine. And it sort of has a hierarchy in the way it looks, depending on what stage of the cell cycle we're at. And so typically when you think of a chromosome, so the DNA of the organism or the cell, we'll just say, it houses all of the genes for that cell. Every single gene, whether it's being expressed or not, is in that DNA. And we think of DNA in these sort of chromosomes, these condensed, packed chromosomes, like when you look at the 23andMe commercials, and they sort of represent it this way. And that's all good and well, but that's really only a portion of the cell's life that it exists this way, because this is a nice way to segregate DNA between dividing cells. It makes it nice and compact. You can easily maneuver it around, and so nothing gets lost. And that differs then from this sort of unwinding organization. We have these nucleosomes and histones. These are just proteins which wind up the DNA. And as we get further and further along, we now see what we're calling a gene. And this is a segment of the DNA which encodes for typically some sort of protein. And as we go further and further, we now are down to those bases. And so this is where like the real chemistry of DNA is occurring. This winds up looking like this if you put it in a cartoon. And so we're seeing these common phosphate backbones. These are what they call the sugar backbone of the DNA molecule. And this is the sugar here, this is the phosphate, the sugar, and then these nitrogenous bases. And it is in these bases and the ordering of these bases that literally encodes who you are. Everything about you and what makes you a human versus a cat versus I don't know, anything, a crocodile, anything on this planet, a tree. The ordering of these bases and what these encode for makes up that organism. I think that's actually really neat. And there's only four of them, but the combination of these over hundreds of thousands of nucleotides. I mean, it can range from a few thousand nucleotides to hundreds of thousands to millions of nucleotides. And yeah, just the different combination that you can come up with makes you who you are. And so sometimes when we hear in the news, we're talking to tie this in with what's happening recently, we'll hear somebody say that, you know, a virus has mutated. What that can mean is, looking back on this, we'll just use this as sort of an example. It's not an exact analogy, but let's say this was the virus. And we know that along this string of nucleotides here, any one of these could mutate, and it can go from a G to an A instead. Like depicted here. These complementary base, T goes to A, G goes to C. But let's say the virus mutates, and instead of putting a G here, it puts a T in this position. So the complement to T is going to be A on this side. And so that's a mutation. That's a single point polymorphism, and it makes it different than every other one that was out there. And so that could mean bad things, but more often than not, it means absolutely nothing. It could be in a region of that genome which doesn't even code for protein. Not every single part of the genome is encoding for a protein. And so I don't want to go into the details on that right now, but just take it for now that just because you hear people saying that it's mutated, it really doesn't mean jack. What's the mutation? Is it stable? Is it in the population and it's causing some difference in a phenotype or a disease? Then we can maybe start to ask questions about what it does. But every single thing on this planet mutates, whether it's alive or it's not, as far as if it has a genome. so RNA or DNA. So just because something mutates doesn't mean it's bad, and it shouldn't be this like point of fear. All right, so I think that's where I'll leave it for now. The next time I want to cover in depth what transcription and translation are, and then what DNA synthesis and replication looks like. Um, and so I think that's a really important process. Again, this is like what blew my mind. This is how we know why things work the way that they do. And even if you only stuck it out for this lecture and the next one and got some information out of this, I think, yeah, I don't know, I think it's really beneficial. And you can start to question some of the things that you hear or people say. And I hope that that continues to be the case as I move on to some of these other review topics like the chemistry and biochemistry one. So yeah, thank you for listening, and I'll try to post some more as I have time.